Hello students, very happy to meet you all today. Today in this session we are going to discuss about the class 12 CBSE biology portions, okay, and here we are going to have a quick revision. So within 30 minutes we are going to revise all the chapters, and after that I am going to tell you some important questions and also the important diagrams which we have to practice. Shall we start? Yes. So these are the things which we are going to see today. As I have told you, the important questions in all the chapters, okay, and apart from that the important diagrams, and I am going to tell you how you have to practice the sample papers and the previous year question papers, okay. Yeah, let's see one by one. So the first chapter, actually even reproduction in organism is there, but that is deleted for CBSE. So we will start with sexual reproduction in flowering plants, okay. So in this the important questions are, the first question is male and female reproductive structure, okay, male and female reproductive structure. Even you can draw a diagram like this, okay. If they are asking you to draw a structure of flower, you can draw the structure of flower and you can explain everything, okay, the stigma, the style and the ovary. And apart from that, these are the stamen. The stamen comprises of anther and filament. So you can see the anther here and the filament here. So stigma, style, ovary here, okay. So like that you have to mark the parts also. Fine. So male and female reproductive structure. When you are going to start a paragraph question, say for example a 3-mark question or 2-mark question, if they are asking you to explain the male or the female reproductive structure, just try to give some side headings, okay. If it is male reproductive structure, you can say andrecium. So you can tell about the stamen, a whorl of stamen will contain the andrecium. Then talk about the anther, okay. You can see the anther is bilobed anther, okay. It is bilobed and tetrasporangiate anther. And when you are talking about the filament, you can say the filament is attached to the thalamus. So like that one line about each and every topic, okay. Then when they are asking you about the female reproductive structure, you can say the stigma. So the stigma is the place where the pollen will come and attach, and the style where the pollen tube, okay, will just enter, and the ovary where the ovules will be present, and these ovules will be converted into seeds after the fertilization. That's it, very simple, isn't it? Then the next one is fertilization in plants. So if they are asking you about the fertilization in plants, you can just simply explain, okay. So this is stigma, style and the ovary, and you can say the pollen grain will come here, okay, and then it will divide, then the pollen tube will form. So here we have the embryo sac. This embryo sac is, okay, 7-celled 8-nucleated stage embryo. That you should mention when you are talking about the fertilization. You have to mention about how the embryo, okay, and how the pollen cell. The pollen cell 3-nucleated stage, and this is 7-celled and 8-nucleated stage, okay. So this will slowly come and meet here, okay. Then the male gamete, out of the 3, 2 male gametes will come and one will come and fuse here with the egg cell to become embryo. That is one fertilization. And the other male gamete will fuse in the center, okay. Center we will call that as polar nuclei. Now it is PEN, primary endosperm nucleus. After fertilization, that will develop into endosperm. So this will develop into embryo. So like that you have to mention, okay. So we have learnt everything detail. So if you want to know, you can just go and see our previous lectures also, if time permits. Otherwise you can just have a quick revision, okay. Then the next one is pollination. When you are talking about the pollination, you have to mention about, like what are the various types. If it is through wind, okay, you will call it as anemophily, and water, you will call it as hydrophily. Okay. So wind and water, and what are the examples of these things, and what will be the specific characters if it is through wind. Okay. So if it is through wind, wind is going to pollinate, the pollen grains should be non-sticky. Okay. If water is going to pollinate, then the pollen grains have to be covered with a mucilaginous coat. And zoophily or zoophily, here maybe if it is with insects, you can call that as entomophily. Okay. Then the pollen grains have to be sticky. So like that you can mention. Then structure of anatrophous ovule. Okay, I have seen in many question papers they have asked this question, a structure of a ovule. Okay. So this picture, when they are asking you to explain that, please draw a picture and label the parts, then definitely they know that you understood the concept completely, thoroughly. Okay. So here you can see these two are the outer integument. Okay, these two are the outer integument. First just try to draw that, then slowly this is nucellus. Okay, nucellus, and inside that is the embryo sac, which has 1, 2, 3, 4, 5, 6, 7 cells and 8 nucleus. And let's explain these things in detail. These 3 cells are called as antipodal cell, and this is the polar nuclei, and this one is the synergid, and the center one is the egg. Okay. So like that you have to mention. That is another important question. Just explain the structure, draw the picture, label the parts. That is enough for you to get good marks. Okay. Then the next question, the important question is parthenocarpy. What is parthenocarpy? Development of fruit without fertilization. In 1 marks you can expect these type of question. And what is apomixis? They will definitely ask this question in 2 mark or 3 marks. Apomixis is the formation or the development of seed without fertilization is called what? Apomixis. And even you can say the advantages of apomixis. If we are developing a seed, apomictic seed, the characters won't be segregated. Okay. Otherwise, to create a hybrid variety, it's very costly. We have to spend a lot, and there is a chance that your desirable character will be segregated. But when you are preparing the apomictic seed, definitely the character won't be segregated, and it is less costly also. Okay. So that's it. We have completed 1 chapter. Then the second chapter is human reproduction. So here again the important question is male and female reproductive system. When they are asking you about the male reproductive system, you can just split it into certain subtopics. Like you can say, first for male, pair of testes. Then go for accessory duct. What are the accessory ducts? And then go for what are the accessory glands. Then finally the external genitalia of male. That's it. Okay. Just try to give these 4 headings, and then you can explain it in detail. That's enough. When you are presenting your paper, the presentation should be very, very clear. Okay. You have to give the side headings and explain each and everything. So when you are talking about the testes, just say about the scrotum also. Okay. Then accessory duct, these are the ducts. For remember the duct, I will give you a trick to remember. RVEV. Okay. Retitestis, vasa efferentia, epididymis, and vas deferens. So you can see here retitestis, vasa efferentia, epididymis, then vas deferens, which is ascending up to the urinary bladder. So that is about the duct. Then when you are talking about the gland, so with this picture itself you can write a 5-mark question. That's what I am saying. You just remember these things and remember this picture, you can write a 5-mark question. So I have told you about the testes, then the accessory duct, then finally we have the accessory glands. So here you can see this is one gland, seminal vesicle, which is the one which is going to contribute more to the semen. Okay, seminal vesicle, then the prostate gland, and finally the bulbourethral gland. Okay, these are the, when you are talking about the glands, you have to mention that, and then the external genitalia, that is the penis, and even you can mention about the foreskin, and the broader part is known as glans penis. Then when you are talking about the female reproductive system, again you can say, ovaries, then you can talk about the accessory duct. So what are the accessory ducts here? And similarly accessory glands and the external genitalia. So if you are going to present your paper like this, then definitely it's really going to be a success. It will definitely give you more marks. So see, if they are asking you female reproductive system, you can just say a pair of ovaries. So you can see the pair of ovaries. Just explain about the ovary. It has outer cortex, inner medulla, like that and all, and it produces estrogen hormone. Just 2 to 3 sentences about the ovary. Then go for ampulla, the ducts, accessory duct. You can see here fallopian tube. This fallopian tube has 3 regions. Remember, again another trick for the regions IAS. Okay, infundibulum, ampulla and isthmus. These are the 3 regions. After that you can see the uterus over here, and then the cervix and vagina. So these are the accessory ducts. Then the glands are mammary gland and vestibular glands. In the vestibular gland it is Bartholin and Skene's gland. Okay, so that you have to remember. Then the external genitalia is mons pubis, clitoris, labia majora, labia minora and the hymen. These are the female structures in the female reproductive system. Then another important question is placenta. So here you should mention about the placenta, the function of the placenta. When you are talking about the placenta, just remember the function of the placenta. Okay, so the placenta, definitely it's going to help the nourishment, and it's going to remove the waste, everything. And apart from that, you have to tell about the hormones which is produced by the placenta. Okay, so HCG hormone, human chorionic gonadotropin hormone, is produced by the placenta, which will help in detecting the pregnancy, and also it will help in increase the life of the corpus luteum. So these are some of the important things which you have to mention about the placenta. If they are asking you to write a short note on placenta, then just tell all the functions of the placenta and how the placenta is formed after the implantation. Okay, so then the next one, this they might ask you in 2-mark question. Okay, what is implantation? So here in this place you can see the egg will come here, when the sperm will attach to it. Okay, that will develop into a zygote. Then slowly the zygote will develop into 2 cell, 4 cell, 8 cell, 16 cell. Then here blastocyst stage, it will come and attach it to the uterus. That is known as implantation. Okay, so that is implantation. Fine. Then embryonic development, how the embryo is developing. So here you can say, students, so as I have told you, this is the zygote. Then from the zygote, 2 cell stage. Then each cell is known as a blastomere. Each daughter cell is known as a blastomere. Then 2 cell, then 4 cell, then 8, like that. After that you can say morula stage. Just remember the stage, draw the picture, explain it in words. Definitely you will get full marks. Okay, then you can say about after the morula stage, you can say about the blastula stage, where some of the cells will move towards the periphery. Okay, don't forget to put the nucleus also. And then here you can see there are, this is ICM, inner cell mass, and this is going to be developed into the embryo. Then you can talk about the gastrulation stage, where 3 layers will be formed. ectoderm, mesoderm and endoderm layers. Okay, these three layers will help in forming different organs, that thereby the organ development and the embryo will also develop. So like that you can explain this question. And another important question is parturition and lactation. Okay, what do you mean by parturition? Childbirth, isn't it? Childbirth. So for the childbirth, the first step is the fully developed fetus and the placenta will send a signal to the mother's pituitary. Thereby, the second step, the mother's pituitary will send a hormone called oxytocin hormone. That oxytocin hormone will contract the uterus. Thereby, will contract the uterus and that will result in parturition. Okay, like that you can explain. Then about the lactation, you can say, the HPL is helping human placenta, lactogen and even oxytocin helping to let down the milk, and the first milk is colostrum which contains IgA antibody that will help in the immunity for the baby for first 6 months. So like that you can explain. Then menstrual cycle, when you are talking about this is again another important question, sometimes you might get this in case study also. Okay, here you should mention about the 4 phases. First phase is menstrual phase. Menstrual phase, how many days? From 1 to 5 days. Okay, then the next phase is follicular phase. Okay, in the follicular phase the follicles will start grow. Okay, that is from 6th to 13th day. Then ovulatory phase, 14th day, ovulation occur. On that day the LH hormone will also be high. Okay, then the last phase is the luteal phase or secretory phase, where the corpus luteum will secrete the progesterone, and that will last for from the 15th day till 28th day. Okay, that will result in the next menstrual cycle. If the female is pregnant, then menstrual cycle won't occur in them. Okay, like that you can mention. So please put side headings and then mention about each topic. So these are the important questions. Then in the reproductive health you have to they won't ask you to explain about birth control methods in 5 marks and all, but in 1 marks you can expect what are the various types of methods. Okay, barrier method and natural method. Lactational amenorrhea is the natural method, barrier method, male condoms, female condoms, like that. Hormonal, IUDs, intrauterine devices, what are the various types of IUDs, hormonal IUD, and then Lippis loop, like that, copper T. So many things. Okay, so you have to learn about all these methods, and the permanent method is vasectomy in male and tubectomy in female. Then STDs, sexually transmitted diseases. So you can learn about gonorrhea and the causative organism Neisseria gonorrhoeae. Okay, like that you have to learn about the diseases, the causative organism and the main symptoms. Only 4 or 5 diseases are given in our book. You can just mention that, that is more than enough. Okay, just learn about syphilis, gonorrhea, and then HIV, okay, and HPV, human papilloma virus, genital herpes, like that. You can expand this question. And then ART, very important question. Okay, assisted reproductive techniques and the significance of reproductive health. This you can write in your own words, why the person should know about the reproductive health, like that, and all you can say. Okay, these are some of the reasons, just to avoid abortions and all. They have to learn about this, just to know about the hygiene, like that. You can develop your own thing. But for ARTs, there are many types of ARTs, isn't it? So the first one is IVF, in vitro fertilization. In vitro means outside the body. The fertilization will occur outside the body, and then ET, the embryo will be transferred into the uterus. Then the next one is GIFT, gamete intrafallopian transfer. This can be done to the female who couldn't produce the egg on her own. Okay, that is GIFT. And then the gamete can be transferred here, that is the egg can be transmitted from another healthy female. Then ZIFT, zygote intrafallopian transfer. Okay, zygote intrafallopian transfer. Here the blastomere should be, less than 8 blastomere stage, not over then. Okay, less than 8 blastomere stage only we can, put the thing in the fallopian tube. After 8 cell stage, it has to be implanted in the uterus. Okay, IVF, GIFT, ZIFT, then ICSI, intracytoplasmic sperm injection. In the egg, they will directly insert the sperm. Okay, cytoplasm of the egg, they will directly insert the sperm, ICSI. Then the last one is AI, artificial insemination. Will be done for the the male or the husband who is having a less sperm count. For them, they will do this procedure, artificially inseminated. Okay, so like that you can mention. Then the next topic is evolution. Here you have to tell about, homology and analogy. Homology is divergent evolution. Which means same origin, okay, same origin, different function. Say for example, forelimb, okay, forelimb of a cat, it is helping it to walk, isn't it? Forelimb of a bat, the origin is same, mammal, but the bat, it is going to help in flying. And forelimb of a human being, we are using it for writing, grasping something and all. Okay, so like that, same origin, different function. Analogy, rhythmically remember, and okay, convergent evolution, different origin is different, but the function is same. Okay, say for example, insect, different origin from the phylum Arthropoda, but it is flying. Bat, mammal, flying. Bird, aves, flying, isn't it? And even you can say pterodactyl, reptile, flying. So like that you can say. Then this is another important question, Hardy-Weinberg's principle. When you are talking about the Hardy-Weinberg principle, just talk about what is a gene pool. Okay, if they are asking it in 5-mark question, then start with what is the gene pool. Okay, then the equation p square plus q square is equal to 1. Okay, so let me just, okay. So when you are talking about the Hardy-Weinberg principle, just talk about the gene pool. Okay, so in the gene pool we have capital A, capital A. This will be represented with small p, and capital A, small a, capital A, small a, 2pq, and small a, q square. So the equation will be p square plus 2pq plus q square is equal to 1. So even you can write this as p plus q the whole square is equal to 1, or p plus q is equal to 1. Okay, sometimes in problem type of question they will give you the p value and they will ask you to find out the q value or the 2pq heterozygous condition value. Okay, then even you can say about the factors affecting Hardy-Weinberg principle, the gene migration, the mutation, the genetic drift. Okay, so like that you can say, and the recombination, natural selection, these are some of the factors which is affecting the Hardy-Weinberg. Then you can talk about the mutation also. Okay, a sudden change, genetic drift. What is genetic drift? A sudden change, isn't it? A sudden change in the environment that result. Say for example, okay, so here in the bottle, this is also known as bottleneck phenomenon. Here in the bottle we have two types of population. One is like this and another one is like this. And due to some circumstances, okay, some unfavorable conditions, only very few of this is going to, whichever one is having the variation, that is going to the next generation, and these things are the founders of next generation. Okay, that's why this is also known as founders principle or Sewage-Wright effect. Then after some time you can see the generation, like this, with more stars population. Okay, so like that is genetic drift. Then theories of evolution is the very, very important one. There are 3 theories. Okay, they might ask you any one. So theories of evolution, and this I have told you, homology of mammals 4 limbs you can see. Then analogy of wings. Then Lamarck, these are the 4 postulates. Internal vital force, then effect of environment and doctrine of appetency, use and disuse, then inheritance of the acquired character. You can remember like this. Okay, a giraffe is there, it has the internal vital force that my neck has to grow, because then only I can survive, I can feed the leaves, which is present above. So that is the internal vital force, and like if it is eating, then only it can survive. Then if it is using it again and again, it will grow, and if it is not using it, it won't grow, and if it is getting that, this character will be inherited to the next generation. Okay, then these are some of the examples proving the Lamarck theory. This is disuse and that is use. Then features of Darwin's theory: rapid multiplication, limited food and space, struggle for existence, survival of fittest. Okay, so whoever is having variation, they will survive. Then the nature will select the organism which has the variation, and this character will be inherited to the next generation. Then the next theory is mutation theory. The mutations are sudden and inheritable. They are the raw material for evolution, and in naturally breeding population, the mutation occur from time to time. Then they appear in any conceivable direction, because always the mutation won't be advantageous. Sometimes it can be disadvantage also, and sometimes no effect. That's it. Okay, so they are discontinuous and strictly subjected to natural selection. These are the salient features of evolution. Then the next chapter, human health and disease. Here you have to learn about all the common diseases in human, their causative organism and the symptoms. Okay, bacterial, viral, fungal, helminthus, protozoan, like that you can split and just try to remember the names. Okay, if possible, just try to remember the, just write a name of the organism, like name of the disease, the causative organism, the symptom alone. Or else even you can, because for some diseases and all, you all know the symptoms, isn't it? Like typhoid, cholera, this and all we know. But maybe only the disease and the organism, the disease, organism, typhoid, salmonella, typhi, like that you can write and keep it in a place where you can see often. Okay, for then definitely it will help you. Then the next one is structure of antibody. Very, very important question. Okay, you have to know about the structure of antibody. It has H2L2 chain, light chain, and then it has heavy chain. Then in between we have a disulfide bridges, constant region where the antigen is going to attach. That region is variable region, like that and all you have to mention. Then this is, this topic, adolescence, it's a definite question they will ask you, and you can write it in your own words about the adolescence. Okay, what are the changes, the causes, the effects, and some adolescents are addicted to some habits, why they are doing that, what make them to do all those things, and what will be the effect of that, the cause of that and the effect of that, like that you can write. And then the last topic is immunity. So when you are talking about the immunity, you should talk about innate immunity. Innate immunity, there are After innate you can say physical barrier, physiological barrier, cytokine barriers, okay, like that and all you can say about the innate. Then when you are talking about the acquired immunity, just talk about active and passive immunities, okay. Active immunity, passive immunity is giving the antibodies directly, active immunity is giving the antigen, thereby the body will produce the antibody. So like that you can remember, and even antigen antibody interaction, then I have told you about the antibody structures, okay. Cell mediated immunity and humoral mediated immunity. Cell mediated immunity means the cells, usually they will ask this question for graft rejection, which immunity is used, it is cell mediated immunity, okay. Humoral mediated is antibody produced immunity. Then the next chapter, very very important chapter, principle of inheritance. In genetics, definite question, we have already taught many times about the monohybrid cross and the dihybrid cross, just go through that, okay. So when you are writing the crosses, just write it down in steps very 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 clearly, okay. First you have to write about the parents, okay. Say for example in monohybrid cross you will write capital T, small t, then write what this is, okay. Homozygous tall, homozygous recessive. Then after that go for gamete for F1, gamete for F1, okay. Then selfing, so like that you have to write step by step. So selfing we will get, you know, like F1 generation, then parents gamete for F1, then F1 generation, then okay, then gamete for F2. So gamete for F2 equal chance, isn't it? And then write F2 generation in a Punnett square. Okay, then write the phenotypic ratio 3:1, okay, 3 tall, 1 dwarf, like that you have to write. Then finally genotypic ratio 1:2:1, capital T, small t, small t, small t. So like that you have to write everything in steps. Then I try to explain it for both monohybrid and the dihybrid crosses, the ratios, everything, okay. Then learn about the Mendel's laws, 3 laws, okay. And then non-Mendelian inheritance, what is incomplete dominance, okay. Incomplete dominance where both the parents is not going to express themselves, red flower, white flower, and we got a pink flower, isn't it? So like that you have to mention. Then codominance, example is AB blood group, both the characters of the parents are expressed. Then multiple allelism, again ABO blood grouping, okay. ABO blood grouping is an example for this. Then pleiotrophy, polygenic inheritance, which means many gene responsible for one character. This is just opposite to this, okay. One gene responsible for many character. So like that you can explain. And then Morgan's experiment, Thomas Hunt Morgan, he did with Drosophila melanogaster, okay. That experiment also you have to remember with the ratios. Then sex determination, okay. How the sex determination in human being, how it is in insect, and how it is in birds, like that and all, XX, XY, XX, X0, ZZ, ZW, like that sex determinations also you have to remember. Then go for genetic disorders. So the disorder you can divide that into Mendelian disorder and chromosomal disorder. Mendelian disorders are the disorders which is going to obey the Mendel, and it is going to occur in the chromosome, okay, in a part of a chromosome. So here you can see hemophilia. Hemophilia is the X-linked recessive disease, okay, X-linked recessive disease, bleeder's disease, okay, it won't clot. Then sickle cell anemia is and the thalassemia, both are autosomal recessive disease, okay. And sickle cell anemia is a qualitative disorder where, okay, in the hemoglobin there are 2 beta chains. In the second beta chain, in the 6th position, instead of glutamic acid, valine will come. That will result in sickle cell anemia. Okay, thalassemia is the quantitative disorder. Then phenylketonuria again is the autosomal recessive disorder. Remember about the enzyme phenylalanine hydroxylase. Okay, if this enzyme is not present, okay, then that will result in phenylketonuria. Even for your 1 mark question, it's very helpful. Then Down syndrome, trisomy 21, the 21st chromosome will be thrice. Then Klinefelter syndrome, actually for a male we will depict XY, isn't it? But here it is XXY. So these male will have a development of breast, everything. And Turner syndrome, normally for female we will say XX, but here it is XO. So the ovaries are rudimentary, they won't develop the well developed ovary. That's it about this chapter. Then the next one is molecular basis. Here you have to mention about the structure of DNA. Okay, when you are talking about the structure of a DNA, just say about the bases. Okay, just talk about the bases, purines, pyrimidines, everything, and then the bonds which is present in between them. Okay, so like that. So hydrogen bond will be present in between the nitrogen bases, and here you can say N-glycosidic linkage, and this is phosphodiester bond, like that. Then another important question is DNA replication, how the DNA is going to replicate or make a copy. Okay, here you have to mention about the enzymes. Helicase enzyme will help in unwinding. Then to remove the tension, topoisomerase is helping. Then DNA dependent DNA polymerase is going to add the nucleotides. Then lead strand will form. Then again lagging strand with Okazaki fragments will form. This will be ligated with the help of DNA ligase, like that you can explain. Then transcription. Transcription is the one in which from the DNA the message is going to be carried to the mRNA. For translation, from the mRNA the message is going to be carried to, with this message protein will be developed. That is translation. We have discussed everything in our previous videos, just go and check it. Okay, these are the important questions I am saying. Then expression of gene, that is lacopron concept. This I will explain it when I am explaining the diagrams. And then HGP, this is just theory part. You should know what is Human Genome Project, okay, and what are the salient features and what are the functions and what are the goals of HGP. Like that you have to prepare this question. And in DNA fingerprinting you have to say the steps. Okay, so transcription I have told you. These are the steps, students: initiation, elongation, termination in prokaryotes. Okay, when you are talking about the eukaryotes, apart from that you won't call this as RNA, you will call this in eukaryotes as hnRNA. After that, this hnRNA will undergo 3 steps: splicing, capping and tailing. Splicing, capping and tailing. So after these 3 steps only you will call this as mRNA. Okay, splicing, methyl guanosine triphosphate will be added. Capping is splicing is the introns will be removed. Okay, introns will be removed. capping is methyl guanosine triphosphate is added. Tailing is polyadenylation at 3' position. Okay, so MgPpp, then the introns are removed and polyadenylation residues will be added. So that is in eukaryotes and this is in prokaryotes. Then 𝗏𝗡𝗄 concept. 𝗏𝗡𝗄, first you should draw this structure. Okay, and then what is this I? This is regulatory protein. Regulatory protein or inhibitor, and this will produce a protein called repressor protein. And this P is the promoter region where the RNA polymerase will come and attach. And this is the operator. And these three together are the structural gene. Then the Z gene is going to code for beta-galactosidase that will hydrolyze the lactose to galactose and glucose. Then Y gene codes for permease that will increase the permeability of the cell to lactose. Then A gene trans-acetylase, it will help in activating the beta-galactosidase. Okay, this is general for 𝗏𝗡𝗄 concept. Hereafter you have to mention about in the medium when glucose is present, switch off mechanism. Switch off means this repressor protein will go and bind to this operator. So all these events won't occur. That is switch off. If glucose is present, then no need for the beta-galactosidase enzyme to break down the lactose. That is switch off mechanism. And if in the medium glucose is not there, then the lactose will bind here, so that it won't go and attach to the operator. If it is not attaching to the operator, then the RNA polymerase will slide over it, and then the Z will do this function, the Y will do this function, and the A will do this function. That is switch on mechanism. Okay. Then the last topic of this chapter is DNA fingerprinting. Just write down all these 6 steps and try to explain each and every step. So the first step is isolation of DNA. Okay. So say for example in the sample in a crime scene, if we are getting a blood or a hair cell or a semen or something like that, we have to isolate the DNA from that. Then digest that DNA using restriction enzyme, then separate it with electrophoresis, then transfer it to the nitrocellulose paper, then hybridize with the DNA probe. Okay, which means probe means these are the complementary strands. Say for example, if this is ATA with radioactive thing is here, ATA, and in your sample, okay, in your, if your, this is the thing which we got from the crime scene, and this one is the suspect's thing. Okay, if the suspect's thing is TAT, then this will come and bind here. All these probe will come and bind here, so that we can say that okay, he is the criminal. The DNA fingerprints matches. That is hybridization. Then deducting this using a radiography, X-ray, okay, autoradiography. That's it. Then organism and population. So here adaptation, this is very easiest chapter, students. Just even when you feel very bored or something, also you can take this chapter and then just learn adaptation, how kangaroo rat is doing the adaptation, concentrating the urine, okay, and fat oxidation is water, the byproduct of fat oxidation is water, and how the desert plants are adapting, CAM pathway. So something like that, you can, this relaxedly you can learn. And even how the desert plants and the behavioral adaptation by the desert lizard, just whenever it feel cold, it will just go and stand near the sun, and if it is very hot, it will just go and burrow, or it will go to the shady place. That's it. These are some of the adaptations. And learn about the growth curve, students. Okay, very, very important. They will ask you these curves. This one is the exponential growth. Here it says, as the time increases, the population density will keep on increase. Okay, but it is unrealistic. So here you can say this one is exponential growth, and this is logistic growth. Initially the growth will be very slower, then it will be accelerated, decelerated, and then asymptote, because as the time goes, the population density won't increase. There are some factors which will inhibit the density, okay, such as predators will be present, or no food, no shelter, something like that, isn't it? That will inhibit, that is the carrying capacity beyond which the growth is not at all possible. Okay, then that's it. Then population density, you should learn about natality, mortality, immigration, emigration, like that and all. And finally, very, very important one, population interactions. Okay, so first one is mutualism. When you are talking about mutualism, both the organism is going to be benefited. And predation, 1 plus and 1 minus. Similarly, parasitism, 1 plus and 1 minus. Okay, and then commensalism. Commensalism, for one it is advantage, and for the other one it is no harm at all. Okay, so you can say egret cattle, the bird, you can see some birds are near the cattles. So as soon as the cattle like feeding it, all the insects will come out, so the bird can just take that and go, isn't it? So that's easy. Instead of digging into the soil and getting the food is very, very difficult. So like that you can say. So that is commensalism. Amensalism, minus for 1 and 0. Say for example an ant, an elephant is stamping the ant. For elephant it is nothing, but for the ant it is harm. So like that you can see all the interaction with the examples. Examples are very, 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 very important, students. It's a definite question. Okay, then the next one is biotechnology, principle and process. Here 4 types of questions. Tools, first you have to learn about the tools. Restriction enzyme, what is a restriction enzyme? Endonucleases. Okay, then the naming, the nomenclature, how they are naming the restriction enzyme, and what is a vector, what are the salient features of vector. Okay, here you have to learn about ORI, then this one selectable marker. Okay, then the cloning sites. Okay, and vectors for cloning in plants and animals. Vectors for cloning in plants is this one. Okay, Agrobacterium tumefaciens, which is the natural genetic engineer. Okay, then in animals you can say retroviruses. Like that you have to remember these concepts. Then process, the steps in the rDNA technology. Okay, it's again another important question. So here you can talk about all the steps, the isolation, cutting it with the restriction enzymes, electrophoresis. Okay, separate it with electrophoresis, and then insert it into the host, obtaining the foreign gene product. And the last step, downstreaming processing. Definitely they will ask you this in 2 marks or 1 mark question. What is downstreaming processing? Separation and purification of protein, okay, or the product. Clear, students? Then the next chapter is biotechnology applications. Here Bt cotton, Bacillus thuringiensis. Okay, you should know about the Bt cotton. Then RNA interference, silencing the mRNA, double stranded, by making it dsRNA. So when you are learning about the Bt cotton, just learn about the action, the mode of action. It is normally the toxin. The gene is 𝗙-gene. You should know the name of the gene. 𝗙-gene produce a toxic protein known as Bt toxin, and this will enter the gut of an animal, okay, a gut of a lepidopteran or coleopterans, and then the gut is alkaline in nature. This toxin will get activated in the alkaline cavity, okay. Then genetically engineered insulin. So you have to talk about the insulin structure, the A chain, the B chain. A chain has 21 amino acids, B chain amino acids, and then you have to talk about the C chain or the linker chain, C peptide. If this is present, this is just proinsulin, okay. In the active insulin, in between A chain and B chain, you can see disulfide bridges, okay. This is active insulin. So insulin produced from human, that's why this is known as humalin, okay. The name of the company is Eli Lilly, like that you should remember. And then just 2 mark question, what is GMOs? Genetically modified organisms, okay. And then transgenic animals, they might ask you to write a short note on transgenic animals. What are the uses, you know, like the transgenic animals where we are going to modify their gene. The genes are going to be modified in these transgenic animals, and the functions, okay. What are the functions? You can use these transgenic animals to know the gene, contribution of gene to the particular disease, and apart from that, even we can produce the biological products, okay. So like you can mention about the transgenic cow Rosie here, okay, Rosie. And then after this, you know, like you can say the transgenic animals are also very useful for, you know, testing the chemicals, chemical safety testing and vaccine safety testing, okay, like that you can say. Then the next chapter is ecosystem. Here you can talk about ecological succession. Another important question is ecological succession. It is of 2 types, primary succession, secondary succession. And then talk about xerarch succession and then hydrarch succession, okay. So succession, xerarch succession may be in a rock or something like that. Initially the lichens will come, then herbs, shrubs, you know, like and then higher plants, then the climax community, the forest can come. And in the hydrarch succession, you can even learn the stages also. Definitely this can come in 2 mark question. What are the stages in the hydrarch succession? The first one is phytoplankton. You can see a small phytoplanktons will be here, and then the submerged plant stage, then the submerged and the free floating plant, and then the reed swamp stage, then here slowly marsh meadow stage, and finally the scrub stage and the forest, the mesic community, okay. Then the components of ecosystem, definitely it's a easy question. You are learning this, you know, from your 5th, 6th standard itself. And then what is productivity, okay. And here you should know about GPP, gross primary productivity is what, and what is NPP, okay. So NPP is equal to GPP minus R, the respiratory loss. In 1 mark question you can expect this. And then decomposition, another important question. So what are the steps in the decomposition? The first step is fragmentation, okay, fragmentation, then leaching, and then catabolism, humification, mineralization. Just put the side headings and then explain each and everything. Definitely you will get full marks. Then ecological pyramids, here you have to talk about the pyramids, okay, the upright pyramids and the inverted pyramids also. You should remember that the energy pyramid is always upright, okay. Then you have to learn about the nutrient cycles. There are 2 nutrient cycles. So the first one is you know, the phosphorus cycle and the carbon cycle. So here you can see CO2 is in the atmosphere, that will be fixed by the plant with the help of photosynthesis, and then the plant is releasing the oxygen by photosynthesis. Okay, so the animals are taking the oxygen and again it is releasing the carbon dioxide, and even you can see the vehicles are also releasing, and here the carbon dioxide is in the dissolved form, isn't it or not? So like that you can say it in your own words, and if they are asking about carbon cycle, just say what are the reservoirs of carbon. Okay, reservoirs of carbon, then just see this picture and explain it in your own words, how carbon dioxide is coming here and how the carbon dioxide is going to the atmosphere again, like that you can say, and what are the human activities here, isn't it? The forest fire and the vehicles, everything are human activities. Like that you can present your paper. Just don't write, like a paragraph, just give some side headings and then you can present your paper wonderfully. Okay, then the next one is phosphorus cycle. Here also you can say the reservoir of phosphorus, where and all the phosphorus is present in our body. Okay, so like that in our DNA, in our ATP, in our bones, like that you can mention, and then say the rocks, we are getting the phosphorus from the rocks and minerals. If it weathers, it will just get into the soil, and this soil solution will be taken up by the producers, and then these producers will be consumed by the consumers. Then this producers and the consumer will become the detritus, then they decompose and then get back to the soil again. So that is the nutrient cycling. Okay, then the next chapter is microbes in human welfare. Here you can talk about microbes, how it is useful in your household, and how these microorganisms are useful in the industries. Okay, in the alcohol industry or in antibiotics, how it is useful, or like chemicals or organic acids, how they are useful. And even you can talk about Monaskis purpureus and then streptokinase enzymes. So everything you have to learn in this topic. Then sewage treatment, very very important topic, and I have a mind map for this sewage treatment, because learning this sewage treatment in a paragraph is definitely it's a tedious process. So you can learn that in a just a mind map like thing. Just see here. So this is the primary effluent. Okay, so the first treatment is primary treatment. That is very easy. Primary treatment is you can just like going to dump all the waste water and remove the sequential filtration, we can remove it. And sedimentation is the gut, isn't it? The soil, the pebbles, that and all we are going to remove. That is primary treatment. Nothing, no biological is involved in there. In the primary treatment, only two steps. One is sequential filtration and the other one is sedimentation. After that we will be getting that effluent, that water. We are going to pass that water into the aeration tank, then constantly agitating it. After constantly agitating it, the microbes will grow into flocks. You can see that here in the picture. Then these flocks will consume the organic matter. Okay, then that will reduce the BOD. So BOD, again another 1 mark question. You can see here BOD, the biochemical oxygen demand. Okay, fine. So after this, some more steps are also there. Then this effluent, okay, this effluent is going to be another settling tank. From there we can supply this to the natural water bodies. So before we are sending it, we can just take a inoculum of this sediment, students, inoculum, and keep it for the next process. And then you till here we have sent that to the aerobic tank. Now we are going to send this sludge. Now this sludge is known as activated sludge, because this sludge contains what? Flocks, isn't it? So that's why this sludge is known as activated sludge. Okay, then this is going to be, we are going to send this to the anaerobic sludge tank, and there we will get the, from there we will get the biogas. And even that sludge, after getting the biogas, you can use that sludge as a biofertilizer. So I have given a trick to remember that. And then you have to learn about biogas. Okay, biogas, about the tanks, how the biogas is produced, everything. Then biofertilizers, then biocontrol agents, trichoderma, Bt cotton. Bt cotton you would have already learnt in biotechnology, and you are going to learn that here also about the trichoderma. Then 𝗔𝘀𝗽𝗓𝗏𝗧. This is a baculovirus, a specific type of virus which will help in just killing a specific arthropods, okay, specific arthropods, not other organisms. So when you are talking about the biofertilizers, you can talk about the Anabaena, Nosta, 𝗡𝗃𝗧, cyanobacteria, everything, and how they are useful in useful as biofertilizers. Okay, that's it, the easiest chapter, students. And the biogas plant, you can say the dung and the water in this feed slurry tank, it is coming to the digester, then this is anaerobic, then the gas is produced. This can be connected to our gas or lighting purposes, and then the remaining sludge will be collected here and we can use it as a biofertilizer. Okay, so like that you can remember the things. Then the next chapter is biodiversity. So here you should know about the patterns of diversity. Okay, latitudinal and species area relationship and the equation also. Then laws of biodiversity. Here you should know the causes. What are the causes for the laws of biodiversity? Okay, that is the main thing. So what is the first cause? Habitat loss, isn't it? Habitat loss, alien species invasion, over exploitation, and then co-extinction. These are the four things. You can explain that in detail. Then conservation of biodiversity. So here you should tell about narrowly utilitarian value, broadly utilitarian value, ethical values. If they are asking you to write a short note, then just write all these values and then even talk about in situ conservation and its example, and then ex situ conservation and its example. Okay, in situ Sacred Groves National Park, like that. Ex situ conservation, seed bank, zoological park, botanical gardens, like that you can say. Okay, and for this I have told you, isn't it? For the pattern, the species area relationship, this equation is very, very important. Okay, here species richness, here actually as the area increases, the species richness will also increase, but it is only up to a certain limit. After that it will turn, the curve will turn into rectangular hyperbola, and this equation is important. And in this equation, S stands for species richness, and then A stands for area, Z is the slope of line, and C is the y-intercept, like that you have to written. Normally this values will be from 0.1 to 0.2. Then, so with this we have covered the entire chapters and the important questions in each and every chapter, and in between I have given some tricks on how you have to present the paper also. Now the next thing is important diagrams. Okay, like mostly they won't ask you to draw a labeled structure, draw a neat structure and label the parts and all, but they might ask you certain 5-mark questions. In that, if you are drawing this diagram, it will just help you to get more marks. Okay, and apart from that, why we have to learn the diagram? In certain case study type of question, they will just put A, B, C, D. What is D and E? Okay, if E is not present, what will happen? Or after the fertilization, what will happen to A? So like that and all, they will ask some questions. So for that, learning the diagrams is also very, very important. Okay, just learn these things, students. So you can see the mature, how the mature embryo sac, and this is the antipodal, polar nucleus, central cell, egg, synergids. Okay, just the diagrams, no need for any explanation here. And here you can see, this is for fertilization. You can see the pollen tube is slowly growing and it reaches here near the embryo sac, and one will fuse here, and the other will fuse here. This will develop into pen and this will develop into embryo after fertilization. Okay, then structure of ovule is also very important. Just draw the integument, then nucellus, then embryo sac and the cells. Then don't forget to mention about the hilum and then the funicle where it attaches to the placenta, and then the micropyle region. Opposite to the micropyle region is the Chalasa region. So these are some of the important diagrams. And then in the human reproduction, which I have already told you, if they are asking you to draw the diagrams like the entire male reproductive system now, just draw the diagrams simply. Okay. Then spermatogenesis and oogenesis, again it is very important. We can't say, you know, in a diagrammatic point of view, but this is also important, isn't it? The schematic representation, oogenesis and spermatogenesis is very important. So you can see this is multiplication phase. Okay, then just the growth phase. After that maturation phase, meiosis 1 and meiosis 2, and this is the polar body. So this is again important one. Then spermatogenesis, again multiplication phase, then growth phase. Okay, just the cytoplasmic content will increase, and the maturation phase, meiosis 1, okay, NN, and meiosis 2, 4 Ns. The release of sperm from the seminiferous tubule is known as spermiation. So this also you should see. Okay. Then structure of sperm. So here you can see the acrosome, then the nucleus, the neck part. You can see many mitochondria here which will help in motility. Then the tail, which contains the, you know, center filament with cytoplasm in it. Then reproductive health, actually they won't ask you to draw the diagram, maybe they will ask you what procedure is this. Okay, so you can say this is tubectomy and this is vasectomy. Okay, birth control methods in vasectomy, like that. Then in the molecular basis, this we have already discussed. Transcription, initiation. So what process is this in case study type of questions? This is initiation. Okay, this phase is elongation, and rho factor is there, which means this phase is termination, like that you can say. And this one I have already explained. Lac operon, I, what is meant for this? I, regulatory gene, P, promoter, O, operator, 𝗔𝘀𝗽𝗍, or the structural genes. Okay. Then in human health and disease, they might ask you this, okay, the structure of the antibody, and here you can see the variable region, and this is the constant region, disulfide bridges, light chain, heavy chain, like that and all you can explain. Okay. Then biogas plant. So this tank is feed slurry tank, and this is the digester tank. Maybe they will ask you A, what is this? B, C, D, like that. Okay, if they are asking you in 5 marks, you can simply draw this also. Okay. Then PCR technique, in biotechnology, PCR technique. So here you can see double stranded DNA, then denaturation, it is splitting. Then the next, this is again an important question, students, very important question, PCR technique. Okay, what are the 3 steps? Denaturation, annealing, and here the extension. Mention about the Taq polymerase also. Taq polymerase enzyme, which is helping in extension. Okay. Then carbon cycle and phosphorus cycle in ecosystem. These are the 2 important diagrams, and here also they will ask you A, what is this? Schematic representation, C, D, like that you have to tell. Okay. So, and then next step, what you have to do is just refer the sample papers. By this time definitely you would have at least 5 sample papers in your hand. Just go through that sample papers. Okay, and the previous year question paper also like I'm not asking you to just write down each and every sample paper as a test, because we don't have time right now. Since it's a quick revision, you can just take the sample paper and just say, okay, for this is the answer, for this is the answer, for this is the answer, because now you have learnt everything and you have revised everything and you know what are the questions which is important, everything, isn't it? So you can just go through the sample paper like that. Okay, this I know, this. Sometimes if you didn't know this, just close that sample paper and go refer your book, learn it, and then come back to the sample paper again. So like that, at least try to solve 5 to 6, 7 sample papers and then go for PYQs. I won't say that they will definitely ask whatever is there in the PYQ, but if you are learning the previous year question papers, you will get an idea how the questions were, and even what are the questions which is very, very important also. Okay, that's why I'm just like insisting you to learn some previous year question paper as soon as you are revising, because for our examination we have 3 or 4 days left. In that days, for 2 days you can just revise the important concepts, and then for another day you can give it for sample paper and PYQs, and the final day the diagrams and all those things you can revise it again. Okay, so please give time for sample papers and learning about the previous year question paper also. I am not asking you to sit and write everything, maybe one paper you can just sit and write and see where and all you are making mistakes, and like how you have to be faster. So all those details you can do. Okay, then that's it. So with this we have completed this entire quick revision, isn't it? So within 1 hour we have completed this entire revision, and we have saw what are the important diagrams that we have to remember, how to label the diagrams and how to present a paper, everything, isn't it? So I wish you all the very best, students. Please like when you are writing the writing it in speed, just remember about the accuracy also. Just learn the question properly and then try to write the answer. Okay, yeah. Thank you for watching this session. All the very best, students.